First of all, I would like to welcome everybody, also the audience, to join us today. And therefore, I open officially this academic ceremony in which Mr. Gabriele Obino will defend his academic thesis entitled Bioinspired by Nature, Designed for Flow, Engineering Functional Interfaces for Small Caliber Vascular Grafts. May I invite you, Mr. Candidate, to present a summary of your studies and the conclusion of your thesis in the next 15 minutes.
Thank you, Prorector. Dear member of the committee, dear family, friends and colleagues, thank you all for being here today in presence and online. In the next 15 minutes, I would like to give you an overview of what was my PhD journey. In these years, I focused my attention on the fabrication of functional small caliber vascular grafts. And I would like to start with a simple but striking fact. Cardiovascular diseases are the leading cause of death in the world. And this is mostly caused by the blockage of vessels. And to restore the blood flow, cardiologists often need to place a bypass. And for doing this, they can take an artery from the chest of the patient or a vein from the leg. And this is what we call autologous graft. And the autologous graft works quite well, but there is a problem. Not all patients have suitable vessels available. For this reason, surgeons need to find a solution, and they have very limited alternatives. For large caliber vessels, we have Dacron and Gore-Tex that are two materials that work quite well for big vessels like the aorta. However, when we need to replace a small caliber vessel, these materials fail, mostly due to the clot formation inside the vessel. Because of this major limitation, research focuses on finding a solution using tissue engineering. And here the idea is to have a temporary artificial vessel that we will call scaffold that should guide the regeneration of the vessel itself. So it should need to provide enough support and allow the cells to attach, grow, and regenerate the vessel, with the final aim to have a native vessel that will take place instead of the scaffold. And this is extremely challenging because an ideal vascular graft must satisfy many requirements at the same time. You need to be strong enough for the suture, you need to be flexible for match the vessel's flexibility, and you need to be porous enough to allow cells infiltration, but not too much because we want to avoid the leaking of the blood. And finally, it must provide a surface that is preventing the blood clot formation. In a native and healthy endothelium, the endothelial cells are the cells that are going to prevent this clot formation. But when we have a damaged vessel, the proteins under the cells are able to get in contact with the platelets, and these platelets will get activated, and there will be a formation of the clot. Something similar happens when we put in contact the blood with the biomaterial. Some proteins may attach on the surface of the material in a non-specific way, and this may lead to the platelet activation and the clot formation. And this is something that we wanted to avoid. And during the work that we did for the PhD, we worked on finding some molecules that were able to prevent this clot formation, or increasing the cell colonization on the surface of the scaffold, creating this monolayer of cells that will prevent the clot formation, or having a material that, even without cells, was able to prevent the clot formation and the platelet activation. So we ask ourselves, how can we design such a small caliber vascular graft? And I would like to think that the main idea that we had in mind was to learn and get inspired by nature. I'm saying this because we used mostly natural molecules, and we combine them with vascular graft that we design to mimic the multi-layer structure of native vessels. This process happened gradually during the PhD. And in chapter 3, we purified the marine polysaccharides, and we used the more promising for improving biomaterial blood compatibility and cell colonization. More in details for these chapters, we purified the marine polysaccharides from a sea cucumber and a marine sponge in chapter 3, and in chapter 4, we used these polysaccharides to improving the blood compatibility of polycaprolactone that can be abbreviated as PCL. PCL is a common biomaterial used in regenerative medicine. And when we combined the marine polysaccharides with the PCL, we were able to have a surface that was promoting and accelerating the endothelial colonization, creating this nice monolayer that will prevent the clot formation because it's going to give a natural barrier. And we also obtained a biomaterial that was able to prevent the clot formation even without cells. About the cell colonization, from these images, we can see that the biomaterial without cells had a coverage that wasn't complete on the surface of the scaffold. And these gaps between cells are very important because these areas are the areas where the clot may form. And this is something we wanted to avoid. When we added the marine polysaccharides to this biomaterial, instead, we obtained a full coverage within a few days. And this is what we wanted to avoid the clot formation. Another compatibility that we had to check was the blood compatibility. For doing this, we used the plasma isolated from the blood, and we incubated the scaffolds and the different materials with this plasma. After the incubation of the plasma on the biomaterials, we were able to record a longer time for the blood coagulation of the plasma that was incubated with the scaffold containing the marine polysaccharides. Another important factor was the platelet adhesion on this surface. We wanted to avoid the platelet adhesion. And in these pictures, the platelets are stained in blue. And in the material without the marine polysaccharides, we recorded a high number of platelets attaching on the surface. However, when we use the marine polysaccharides in combination to the biomaterial, the number decreased extremely. Concluding for chapter 3 and 4, I hope I convinced you enough that marine polysaccharides were able to increase the endothelial cells colonization of the scaffolds, but also that these marine polysaccharides were able to improve the biomaterial blood compatibility, increasing the time needed for the plasma to coagulate and decreasing the number of platelets attaching on the surface even without cells. Moving forward, in chapter 5, we use the biomaterial that we optimized in chapter 3 and 4 to build a multi-layer vascular graft. In detail, in this chapter, we combine two different biofabrication techniques, electrospinning for producing nanofibers and a 4-axis printing system for fabricating microfibers. In the multi-layer construct, the first layer was composed of aligned nanofibers in the direction of the blood flow. The first middle layer was composed by circumferential aligned nanofibers, and the second middle layer was composed by circumferential aligned microfibers. These microfibers were also having a higher porosity between them, and this was important for allow cell infiltration, specifically in the middle layer. And then there was also an external layer with random nanofibers, and this was to mimic the external layer of native vessels. An important characteristic that we wanted for an artificial blood vessel was to withstand the physiological pressure of the blood without leaking or bursting. For this reason, we ran the test that you can see in this video, where we increase slowly the pressure inside the graft until we reach the burst. Once we reached the burst, we collected the data, and we saw that the pressure needed for bursting were way higher than the physiological pressure, and this allowed us a safe and good margin of work. Moreover, this multi-layer scaffold allowed the correct cell orientation in the different layers. In the first layer, we had endothelial cells aligned in the direction of the blood flow, that were following the direction also of the nanofibers. And in the middle layer, we had smooth muscle cells that were able to infiltrate between the fibers. However, blood vessels operate under constant flow. So it was important to check the behavior of endothelial cells also under a dynamic flow. For doing this, we used a peristaltic pump to generate the dynamic flow, and we placed the vascular graft inside this rectangular chamber that you can see indicated by the blue arrow. After 7 days of culture in this dynamic system, we were able to appreciate how marine polysaccharides were having a more mature endothelium on the surface of the lumen of the scaffold compared to the biomaterial without marine polysaccharides, but also compared to the biomaterial containing heparin. And heparin is a common polysaccharide that is used already clinically for avoiding the clot formation of vascular grafts. Finally, in the last experimental chapter, we expanded the spectrum of tested natural molecules, introducing on the biomaterial notogen senoside Fc. Notogen senoside Fc is a small molecule that is purified from the plant Panax notogen sang, and is a known platelet aggregation inhibitor. For this reason, we wanted to introduce it inside the biomaterial to improve endothelialization and reduce platelet adhesion. About the endothelialization, so the colonization of endothelial cells on the surface, we were able to see that the scaffolds containing the small molecules were covering the surface in an almost complete way in a few days, compared to the biomaterial without a small molecule. And also about the platelet adhesion, when we introduced the small molecule, we noticed a decreasing number of platelets adhering on the surface of the scaffold, and this decreasing number of platelets was proportional to the concentration of the small molecules used. In conclusion, I will like to give a final overview where in chapter 3 we purified the different acid polysaccharides from marine organisms, and we selected the more promising ones for using them in chapter 4 to improve the biomaterial blood compatibility and endothelial cells colonization. Moreover, in chapter 5, we used this biomaterial that we optimized in the previous chapter to fabricate a multi-layer vascular graft that was mimicking the different layers of the native vessels. And finally, in chapter 6, we incorporated another natural molecules, notogen senoside Fc, to extend the library of options available for fabricating a functional small caliber vascular graft that was able to prevent the clot formation and improve the cell colonization. This was my last slide, and I would like to thank you, my supervisors, Lorenzo and Antonio, my paranims, and family and friends, and all of you for your attention in presence and online. With this, I would like to give back the word to you, Prorector.
Thank you very much, Mr. Candidate, for your very nice presentation, well detailed, also very scientifically, very detailed presentation. And I think we should start now with your defense. And for the audience, today we have 6 opponents who are going to ask questions to the candidate. 3 of them will be here present, and 3 of them will be online, but you will see them on the screen. But before we start, of course, I also would like to introduce the promotion team. That's the team who guided I would say not doctor, but well, he's not doctor yet, but Mr. Oboni, and for the last 4 or 5 years. And first of all, online, I'm going to Professor Lepeda. He's from the University of Sassari. And in addition, we have here present, on the other side of my table, opposite of me, Professor Moroni from our university. They both guided you in the last years. So without further ado, I would like to start, and I give first the possibility to discuss with you with the chair of the assessment committee, Professor van Grinsven. Professor van Grinsven is professor in regenerative medicine at our university, and I'd like to give him the word.
Thank you very much, Mr. Prorector. Dear candidate, it was such a pleasure to read your book. It's so in-depth, so broad, and really also clinically relevant. Nevertheless, I have, of course, some questions. When we go to chapter 4, you have your very nice scaffold, and then you started testing it for coagulation. You used platelets, but you use it in a static manner. However, in real life, you always have a blood flow, you have shear stress, you have flow stress. Does it make any difference?
Highly esteemed opponent, thank you for the compliment of the thesis, and thank you for your interesting question. Indeed, in a realistic situation, we will have a constant flow that will help us, indeed, to have less platelets adhering. That's also what is changing between large vessels, where we have already materials that are working well because we have a higher flow, compared to small caliber vessels, where the time that the platelets have to adhere is longer. And we tested them in a static way because was what also we see was more reported in literature, but also was like giving us the extreme, more complicated scenario. If the platelets adhering on a static condition were decreased so much, we're expecting that in a flow condition, there will be even more, there will be even less platelets touching on the surface.
Okay, thank you. Kind of in the same way, when you then look at your endothelial cells, you do again very much the same. You have like a huge suspension of endothelial cells, you put it to your scaffold, they sit down and they cover the scaffold in a nice way. However, when you use it in the clinic, you make it an interponate, and then actually the cells need to grow from the sides under shear stress conditions. So how do you compare your system of huge amount of cells up to the surface compared to only a small amount of cells coming from the sides?
For trying to simulate this condition, indeed we did dynamic culture, and we had two options at the beginning: to completely endothelize, create an endothelium on the lumen of the scaffold, and then start in the dynamic culture, or to seed the scaffold with the cells and start the dynamic culture immediately without waiting for a endothelialization of the scaffold. We opted for this second option to try to mimic as much as possible this condition that you just mentioned. And indeed, we didn't have a full, complete endothelialization with the dynamic culture, but also because for technical reason we needed to stop the experiment after 7 days because of the condition of the experimental setup. But we saw a very huge difference between the scaffold containing the marine polysaccharides compared to the polymer alone, or even with the polymer with heparin. Because, for example, if we look at the images at day 1 of the polymer with the heparin and the marine polysaccharides, the conditions are very similar in terms of cells adhesion. However, after 7 days, only the one with marine polysaccharides showed a more mature endothelium, expressing vicadherin and cells that were indeed interacting between them.
Okay, so you say actually, so when you used the flow, it was a little bit less. Again, when I'm going back to the real body, you not only have endothelial cells and platelets, but you also have like erythrocytes, leukocytes, etc., which can then actually occupy those spaces. So how would you envision those cells, this more complex mixture of cells, to interfere with endothelialization, and how did it affect your vessel?
Yes, thank you. This is also very interesting. We didn't experiment that directly, but we discussed about that, and we said that these polysaccharides are highly negative charged. And this is a similar characteristic with heparin. And with heparin, there are different studies that show that is able to bind, stabilize different growth factors, between them VGF, that is known to stimulate the endothelialization and the vascular nectar formation. So we discussed that since also our polysaccharides have this similar negative charge, it's possible that they will be able to attract and create this electrostatic interaction with growth factors that may be able to stimulate the endothelial cells formation. And about the immune cells, they may indeed help or not help the situation, but for having more data about this, we should...
So that is certainly something to continue your research on. When I then look at chapter 4, it worked very well, very nice, you have endothelialization, your coagulation is inhibited. And then I come to chapter 6, and you use another molecule, saponin. So, and you did actually very much similar experiments. So what do you now prefer? Do you prefer your marine polysaccharides or your saponin?
So there is a huge difference between these two kind of approach. I didn't go through that in the presentation for a matter of time, but the polysaccharides are attached on the surface of the scaffold with a covalent bond, so they are not supposed to leave the scaffold. Instead, the saponin is going to be released in a short term. And what we had in the chapter 6 indeed was a solution for the first moments of the implantation of the vascular graft. That is also one of the more critical moments. In this moment, we wanted to avoid the blood coagulation and also, if possible, increase the cell colonization. But indeed, I wouldn't propose the solution in chapter 6 as a final solution, but as another instrument that we have available to combine it with other...
Would you then combine the one in chapter 4 with the one in chapter 6?
It may be a solution indeed.
One small last question. When you then look at your saponin, actually your endothelial layer starts to detach. Is that depending on the concentration, or would you expect when you have your polysaccharides in there that would not happen?
As we said, indeed, it's a temporary effect. And I think the reason why the endothelial cells were adhering in a better way on the surface of the scaffold at the beginning was also because the hydrophilicity was increased. But maybe this hydrophilicity will go back normal once the molecule will be released, and that could be a reason why the endothelium start detaching. But combining it, for example, with the marine polysaccharides, we may have both of the properties and have a better solution.
That sounds excellent. Thank you very much for your answers, and I give back the word to the pro-rector.
Thank you very much. The opposition will now be continued by Professor Pandit. He was also a member of your assessment committee, and he's a professor in biomaterials at the National University of Galway in Ireland. And I welcome him and thanks him that he joins today. And I'd like to give you the word, Professor Pandit.
Thank you, Mr. Pro-Rector. And I'd like to also mention that I also enjoyed reading the thesis of the candidate. So the question for the candidate is in very fundamental nature, and this is related to the first two propositions the candidate has. So the question is, in a very simple word, what does bioinspired really mean? And the context of the question is that the word bioinspired is so widely used in a field that we rarely question what we actually claim to use. So your title of the thesis, Bioinspired by Nature, Designed for Flow, and throughout the work you position marine sulfated polysaccharides as functional analogs of endothelial glycocalyx, but the glycocalyx itself is not static in nature. It's very dynamic in nature, as you know. So when we extract a molecule from a sea cucumber, put it on a polymeric fiber, and we call it bioinspired, what are we truly claiming? So in your view, when does biological inspiration end and useful engineering approximation begin?
Highly esteemed opponent, thank you for the comments on the thesis, and thank you for your question. I hope I heard correctly the question, but you are asking mostly to explain what I mean with bioinspired. related to the polysaccharides choice and the structure itself and the title itself of the thesis. And what I meant here was that we have different chemical options for using on the lumen of the scaffolds to improve the endothelialization and the clot formation. But nature provides us with molecules that were developed under years and years of evolution. And I believe that we may find molecules that may still be quite clinically relevant in species that we still didn't even think about, mostly in the bottom of the ocean. And for Bioinspired, I like to think it in this way because we use mostly natural polymers or small molecules that we isolated or that were isolated from natural plants or marine organisms. And moreover, the Bioinspired is also for the vascular structure of the scaffold because we try to biomimic the different layer of the native vessels with this multi-layer structure. And yes.
So in your view, is it that simplistic, just adding one molecule to the substrate creates that biomimicry that we want?
It's not simple, of course, but the endothelial cells itself have this glycocalyx that is mainly negative charged, and also these polysaccharides may mimic well enough this glycocalyx in terms of negative charge and hydrophilicity. Moreover, not only the glycocalyx of the endothelial cells, but also other molecules that were already used for this kind of vascular graft, like heparin, have a similar function. They are anticoagulant polysaccharides, they are negative charge, and they may attract and stabilize growth factors that may allow a better regeneration of the vessel.
Okay, so if I may ask Prorector another question.
Yes, of course.
So, yeah, when is the scaffold ready for the human being? Or when do you think, your multi-layered PCL construct has, as you showed, has burst pressure that exceed clinical benchmarks. You have promising endothelialization kinetics, and you have a supportive SMC or smooth muscle cell environment. What is the minimum evidence that's required that you want to see in this assembled construct that we can take it to the clinic?
Thank you for this question. The connection was a little bit bad. I'm not sure if I heard completely, so if I'm replying a different question, please stop me. And yeah, for the future, for indeed in using actually this vascular graft, the requirements that I hope they will have is, first of all, they should be resistant enough to few months in an animal model. We should check the immune response against these polysaccharides and also the enzymatic response. Something we think is that the enzymes contained in the human body will not attack this kind of polysaccharides like they could do with heparin, and this would be an advantage.
Yeah, so what specific study would you do to do this?
Yeah, we should move step by step, and the first step will be with some small animals for doing a pilot study, maybe some rats, and implanting the vascular graft on the abdominal aorta. And then ideally we should move to bigger animals like sheep that have a very well-known coagulation system that it will be even more reactive than human body. So if we pass the sheep study, we may be more confident on the results also in human body.
Yeah, and if I may probe this further, a lot of previous grafts have done exactly the same, but have failed.
Yes, but they didn't use the same vascular graft and the same marine polysaccharides.
So how does your construct make it more robust than others?
Yeah, sometimes they failed because the compliance in the body wasn't matching the one of native vessels, and with this construct we had a tunability of the different mechanical properties, and we were proposing also to change the different layers in order to match better what is the compliance. And also, for example, the cell infiltration, we had a selective cell infiltration only in the middle layer, allowing the scaffold to do not have any leaking in the first layers, but then to have a selective infiltration only in the middle layer by the smooth muscle cells.
So your hypothesis around the compliance, which they would not have had before, is your hypothesis that this will work better?
It's one of the many things that I can hypothesize, yes.
Okay, great. Thank you very much.
Thank you very much. Then the opposition will be continued by Professor Rotmans. He also was a member of your assessment committee, and he is the chair of internal medicine and nephrology at Leiden University Medical Center. And I also thank him that he came all the way from Leiden to Maastricht. And I'd like to give you the word.
Thank you, Mr. Prorector, and dear candidate. It's a pleasure to be here today and to discuss your beautiful thesis with you, where you did some excellent work in the field of vascular tissue engineering. I'd like to congratulate your promotion team as well with this achievement. But obviously I also have some critical questions for you. And the first one relates to chapter 2, which is more like a general question about these marine sponges, right? You studied nature and you mentioned evolution. Why do you think these very small organisms would have molecules in their in the organism that have anti-thrombotic properties? Because they don't have blood, they don't have coagulation.
That's a very interesting question, and a highly esteemed opponent. Thank you for the question and for the compliment for the thesis. And this is a very interesting question and a very different perspective of what I had for now. Like, indeed, sponges doesn't have blood. Why should they contain something that is anticoagulant? And I think there is not a relation between these two things. They may have developed this structure for some other reason. Usually the sponges and other marine organisms generated these molecules for defense. Maybe was going to defend somehow from some pathogens. Yeah. And we just had the opportunity to find them and use them for anticoagulant properties.
Yeah, I think you're absolutely right, because indeed they affect cell-cell interactions, right? And then it probably relates a bit to the question of one of the previous opponents, right? Because you studied mainly the interaction with endothelial cells, but these, because of the nature of these molecules, they probably also have an impact on the interaction with other cells in the circulation or in the blood vessel. So that is something indeed for future studies to study to what extent, well, these cell interactions would be affected. And then subsequently, well, you studied in chapter 2, where you gave, in chapter 3, sorry, where you gave an overview of different of these polysaccharides, and you learned a lot about them. And then subsequently, I was thinking, wouldn't it be an easier approach just to chemically engineer a ideal polysaccharide rather than retrieving one from nature?
Thank you for the question. Yes, that will be an option for the future. Like that will also help us to have a better production system and have a less variability on the batch of the polysaccharides.
And how would such a polysaccharide look like, knowing the chemical structure that you've studied so far from the polysaccharides?
Yes, the main difference from some molecules like heparin is the branched structure, which is a focusylation on the side of the main structure. And this branching of the structure may indeed have important difference with heparin, that it is a great molecule for anticoagulation properties, but it has a limitation, like the induced thrombocytopenia, or also the degradation, because we have heparinases that may go on the site of the vascular graft, mostly if immune cells will go there and will release this enzyme. So the anticoagulant properties in that case will be temporary. Instead, in our case, we hope will be lasting longer because of the resistance to human enzymes.
Yeah, so you refer to heparin, and I fully agree that the heparin-induced thrombocytopenia is a downside of heparin because it originates from pigs, right? And wouldn't be the development of antibodies against these polysaccharides, like we see with heparin, be a similar problem with your molecules, right? Because they're also recognized as foreign.
It could be. In literature, there is nothing about that. I had a look for writing the review in chapter 2, but I also didn't find any specific studies on this. So it may be something that we may look and more details.
And so suppose together with your team you designed the ideal polysaccharide with perfect antithrombotic properties, and then I've read your work and also others, right? We really try to mimic a complete biological vessel, but for the applications that you mentioned, like in the heart and in the legs, the only thing that is actually needed is a conduct that flows blood from one side to the other, right? Where there is no occlusion and no thrombosis. Wouldn't it be an easier approach to, well, with the help of chemists, to actually apply these small molecules to a piece of plastic rather than building a complete new blood vessel, which is extremely complicated, as you've shown?
Yes, but this will not. We built this different multi-layer structure, also having a nanostructure that was able to increase the internal surface, allowing a better cell endothelialization compared to a single tube connecting the two components.
But I think you don't even need endothelial cells if you have molecules that do not induce thrombosis.
But what do you mean with molecules that don't induce thrombosis? On the surface or circulating in the blood?
On the surface.
Yes, but there are already studies and clinical application where this vascular conduct doesn't work well enough for a vascular graft under the 5 millimeters. That's why we studied a better way or something that may be better for building functional small caliber vascular grafts.
Okay, thanks a lot, dear candidate. I'm sure we will have some time afterwards and in the future to discuss this more in depth, and I would like to give the word back to the prorector.
Thank you.
Thank you very much. Then the opposition will be continued, now again online, and we go to Dr. Crosio. She was a member also of your assessment committee, and she is affiliated with the Department of Biomedical Sciences at the University of Sassari in Italy, and I would like to welcome her and give her the word. Dr. Crosio.
Thank you, Prorector. Mr. Candidate, I compliment you for the complexity and of your thesis that has a quite logical progression along the different chapters. However, I have few questions for you. And in your thesis, you argue that the marine sulfate polysaccharide are superior in their activity of functionalization of scaffold in terms of support for endothelial cells. But there are already published other alternatives to this surface modification, like surface that can release nitric oxide or scaffold that were immobilized with the BAGF. So how can you compare your approach to these alternatives? And in your opinion, what is the unique advantage of using MSP to use such a complex matrix? Because sea cucumbers are also a protected species. So I'm wondering how you can produce this molecule on large scale.
Esteemed opponent, thank you for your question and for the compliment for the thesis. And I would like to reply saying that indeed there are other solutions. I also mentioned two of them in this presentation. And one was indeed a release, like the release you are talking about the nitric oxide. This has been quite effective indeed for surfaces that need to get in contact with blood, because they're going to indeed release nitric oxide. It is something that endothelial cells do by themselves, preventing part of the platelet activation. So when we release nitric oxide, we have a similar effect on endothelial cells. And moreover, nitric oxide may also stimulate endothelial cells to attach and go there in the site of the vascular graft. However, it's a release. So eventually, after some time, will be lower and lower. Instead, with the marine polysaccharide, we have a covalent bond between the marine polysaccharide and the scaffold itself. And also we have these anticoagulant properties that is definitely stronger than the nitric oxide, because the nitric oxide is just, the release of nitric oxide is just simulating what the endothelial cells will do in part, but it's not the only thing that endothelial cells are going to do. But it's still a very good thing that we can use to combine maybe with other things. Like, for example, I can imagine that after the ship trial, if we don't have enough anticoagulation in the vascular graft, we may, for example, introduce also the use of the other small molecules, like the release of nitric oxide. For the question about how to have these molecules, because of sea cucumber may be protected in some areas, it depends also where you take the sea cucumbers, because, for example, in some Asian market, it's very common also to buy it for just for food. Like you can go and buy the sea cucumber. But we also propose and discuss that it may be possible to synthesize chemically the structure, and having even solved the problem of the variability between the sea cucumbers.
Thank you. And do you think this chemical synthesis is feasible? Do you have any clue about that?
So, I didn't study that directly, but I don't want to go in a field that is not mine. But I may imagine that since it's a fucosylated chondroitin sulfate, we may start from the chondroitin sulfate and then create the branching with the fucose and try to work on that.
Thank you. I have another question from a more biological point of view. Do you think that you are proposing a quite sophisticated biomaterial functionalization strategy to develop the small caliber vascular graft? But do you think that the field will move towards more biological solutions, such as IPS derived endothelial seeds, for example, as endothelial progenitor to recruit in a more physiological way by chemokine gradients the other cell type that will finally constitute the vessel?
Yes, there are different studies indeed using induced pluripotent stem cells for this kind of vascular graft. The idea is to have the graft, seed the cells before the implantation, and implanting the cells, the graft only after reaching a complete endothelialization. The problem here could be, for example, the timing, because if the patient needs to go under a urgent surgery, you don't have the time to get the IPCs and create the vascularization, the vascular layer inside the lumen, and then implant it on the patient. So the timing is one point. And the other point is that Often it happened that when you have these cells seated on the scaffold and you implanted it in the blood flow, these cells may detach quite quickly. Instead, we wanted a graft that was able to recruit cells, endothelial cells, and cover the entire surface of the lumen, but also that was able to prevent the clot formation even without cells. And this is something that we were able to obtain using these marine polysaccharides.
Thank you very much. And now I would like to give back the word to you, Prorector.
Thank you very much. Then the opposition will be continued. Now we go back online to Dr. Wlodarczyk Bigun. She is affiliated with the Department of Polymer Sciences at the Zonnike Institute for Advanced Materials at the University of Groningen, and I would like to thank her and welcome her, and I give her the word.
Thank you, Mr. Rector, for giving me possibility and to talk in the voice. And Mr. Candidate, I would like to firstly congratulate you and also your supervisory team on a beautiful thesis. I really like the way how the story built up and how the complexity also of your material system was building up throughout the whole thesis. And I'm especially, because of my biomedical engineering background, fascinated by the way how you produced the system using different printing approaches. And this is where I would like to ask some questions and clarification and have some discussion. But first of all, reading the introduction, I was thinking about what you said, that for large diameter vessels, usually non-degradable materials are used, whereas for the small ones, you propose to use the degradable ones. And my question is why? Why that would be better, and why it cannot be the other way, so that for the large diameter also the degradable materials are used, and why you propose for the small one, the degradable ones?
Esteemed opponent, thank you for the nice comments and for the question. Ideally, it may be also useful for large caliber vessels, but we didn't focus on them because we have already solutions that works quite well, like the Dacron and the Gore-Tex, that is EPTFE, and it's an excellent material. It's quite repelling all the most of the clot formation, and this works well indeed with larger vessels because there is a higher flow, and the blood proteins and platelets have less time to interact with the material. So we didn't focus on that because since we have a solution that works well, why to continue studying on that? But it could be an option maybe in the future. But also that will require a stronger vascular graft because indeed will be bigger, will be needing different mechanical properties.
Mm-hmm. Yes, I understand. But then why you suggest now for the small one, the degradable one? Is it the degradation kind of additional point that you have within your system, or is it really requirement that for the small ones we need to have a degradable material?
No, if we were able to have a material that is going to avoid completely the blood clotting without any cell colonization, that would be an option. We wanted to have a biodegradable material because we also wanted an integration of the vascular graft in the human body. So in the end, the best vascular graft that we can have is a native vessel. And if we were able to reaching this aim, we will be sure to get a really good vessel.
Mm-hmm. Yes, probably that is also the reason why maybe still the big ones, which are non-degradable, could be degradable, that this would be still improvement, and that would be the answer to your question, why to change something that is still working. But then I would like to switch now to chapter 4, and then in figure 4.2 you showed how your material coating is influencing water contact angle. And then you mentioned also in the text that mostly polycaprolactone is regarding hydrophobic, but in principle the bulk material, or when you have just a flat surface of bulk PCL, the contact angle will range between 70 to 90. So actually it is slightly hydrophilic. And it seems, or it is what it is, that you are changing the hydrophobicity, or you are inducing it, or even the super hydrophobicity, what we can see in figure 4.2, by the design, right? So I would like to see your point on that, and if you think that also by changing the design, you could change the properties of your vessels.
Yes, for sure, the water contact angle may change according to the structure, and in our electrospan scaffolds, we indeed recorded a very high contact angle, above 120, and this is what we can call super hydrophobicity. And it's also what I saw reported in literature, so I didn't focus too much on the structure itself, also because the final aim was to use these marine polysaccharides that itself were decreasing the contact angle immediately so much. And yeah, we didn't think about changing the structure to changing the water contact angle, because the hydrophilicity wasn't the only problem of PCL. The bigger problem was that is a non-specific adhesion of proteins on the surface of the scaffold that then was giving a denaturation of the proteins, and finally the platelets will activate because of this denaturation of the proteins on the PCL. That will happen even if the contact angle will be a little bit lower.
Mm-hmm. Okay, thank you. I think I don't have too much time, but then I would like to go also to the question that it's super interesting for me. How actually did you handle the process of doing this 4-layer scaffold? Because what I understood but could not still imagine very clearly is that you were actually changing the printing stage. So it was not always on the mandrel, and then you have different fiber alignments and different techniques you are using. Could you explain how you were handling this material?
Yes, indeed we were changing the orientation of the stage. So the first layer was basically aligned nanofibers, and we actually roll it on a mandrel, following the direction we wanted. The second layer was the same approach, and that's also why it's so important that the third layer was printed by the 4-axis printing system, because we had only 2 layers that were adhering to themselves, but they weren't completely an homogeneous structure. So at this point, we moved the mandrel in the 4-axis printing system, and we printed the microfibers that were giving a better structure at the construct. And with these fibers, also the underneath layers were getting more homogeneous. Then we moved the mandrel on the electrospinning machine for creating the last random electrospan layers, and this helped us even more to create an homogeneous construct. I don't know if I replied.
And how did you could ensure that there is no
Sorry,
delamination?
I do have to interrupt. We have to proceed to the final opponent. So thank you very much. And we have to proceed to Dr. Bergani. She was also a member of your assessment committee, and she's affiliated with the Department of Complex Tissue Regeneration at our university, and I give her the final opposition.
Thank you. Dear candidate, I also would like to congratulate you and your promotion team for this beautiful piece of work. Your thesis has a very strong element of biomaterials design. And I am a big fan and advocate of biomaterials design, and that is why probably I'm also sitting here today. So I enjoyed it a lot, but I still would like to challenge you in the next few minutes in a number of questions. My first question is overall about the approach that you have in your thesis, and I would like to start that discussion by asking one of your paranames to read the proposition number 8.
More often than not, systems fail not because of missing data, but because critical details are overlooked.
Thank you. Thank you very much. So while this is a quote from someone else, you still included it in your propositions. there are two things in this statement that I do not understand, and I want to see the relation of them to your thesis. So first of all, when you say that critical details are overlooked, does it mean if we take attention to all of the critical details, our approach is guaranteed to succeed?
Dear esteemed opponent, thank you for the compliment and thank you for your question. I wanted to include this proposition because I'm a fan of this writer, and what he meant here is that we can look at many details. We can look and we looked at many details indeed. That could be a bit what you are questioning. But in the end, what matters is if the vessel works. And in this case, I think we demonstrate enough that we had a better cell colonization and a very strong anticoagulation properties due to these molecules that we used. And yeah.
Okay, so the second thing that I did not quite understand from the statement itself is that what are those critical details? I find that phrase actually quite subjective. So in the context of your thesis, what are the critical details that have to be considered?
I wanted to mean here that we paid attention to many details indeed, like the lamination that was asked by the member of the committee before, and the way of fabricating the scaffold, the different layers. So these are the critical details that we were checking, and we were paying a lot of attention in, okay, will the layer detach one to the other? Will the cells actually infiltrate in the scaffold? Will the marine polysaccharide stay there, or they will be removed after the first 30 minutes? And we put ourselves a lot of questions that sometimes were not needed, sometimes yes. And the final important question to be replied is, does this system work? And I hope I convinced you enough that it may work. And this was the...
Yeah, okay. I think I have still time for one question, so I will continue the same discussion, and I would like to ask one of the details that I find important for designing such material, and that is the choice of the base material. So in chapters 4 to 6, you actually used PCl as the main material and then functionalize it. You know that I come from background of bone regeneration, and PCl is actually quite a popular material there. And it's because of mechanical properties, but also how it interacts and whether or not it induces osteogenic differentiation in cells or not. But there are many evidences that show that this material actually mineralizes. So you have chosen this material to put it in a vessel environment where you also have ample amount of different factors that contribute to mineralization. In your opinion, is this something that has to be taken into account, and you should study it for this kind of materials in a vascular system?
Thank you for this question, because allow me to discuss more on the choice of the material. So we selected the material itself in that moment because it was a material that was used quite often also for vascular grafts. And it really depends also on the structure. It can have very different mechanical properties. For what concerns the mineralization, I saw that mostly in the paper where they were reporting this was after different months, and it was because cell infiltration wasn't sufficient. And this is because sometimes when you have an electrospan scaffold, the porosity is very limited, and you end having a kind of sandwich of cells from one side and to the other side. In this case, instead, we wanted to use also the microfibers to allow a higher porosity, and we demonstrated the smooth muscle cells were infiltrating very well in the layers, and also the phenotype was maintained.
Dear Mr. Candidate, there are two options now. You are allowed to finish your answer to our opponent, or you have the freedom to say, I stop.
I will just add one sentence.
Okay.
And also the phenotype of the smooth muscle cells that are the ones that then are responsible in vascular graft to calcify were better maintained with marine polysaccharides compared to the other option that we tested.
Thank you very much. I'm satisfied.
Thank you very much. Dear Mr. Candidate, the time appointed for defending your thesis has now officially passed. The degree committee will now withdraw to discuss the quality of your thesis, but that has already been approved. But we're going to discuss the way you have defended your thesis in the last 45 minutes. And we take also in consideration your presentation. I request that you and your company here present in the audience await the results of our deliberations and the return to the aula. Thank you very much. Dear Mr. Obino, the degree committee here present has assessed not only the quality of your thesis, because it has been approved, but we discussed vividly the way you have defended your thesis, and of course also we took into consideration your presentation. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Moroni is authorized to confer you this academic distinction in accordance with Dutch university customs, and therefore I give the floor now to your promoter.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Gabriele Obino, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university.
And before we start the laudatio, I also would like to mention this is a double degree, a degree between Maastricht University and the University of Sassari. So I also congratulate Professor Lipado, who is, of course, online somewhere available. And I give the word now to Professor Moroni to speak out the laudatio.
Thank you, Mr. Prorector. Dear Dr. Obino, dear Gabriele, as always in life, patience is of key importance, with a lot of patience, of course, but also a lot of passion, perseverance, and resiliency. You have finally come to the end of your doctorate journey. It has been a special pleasure for me to see the man you have grown into, a creative thinker, combined with the right pinch of critical mindset that training into a scientist requires for professional development. We ought to know each other about 3 years ago, roughly speaking, I think. Thanks to Antonio, actually, who had the vision to investigate the conditions to bring you to Maastricht as part of your PhD studies, which became the prestigious double degree doctorate that we are here together gathered for. Gabriele, you made since the beginning the right impression. A young man who emanates calm, positivity, and a strong will. The beginning was tough, not just a totally new environment to adjust to, but also the perils of some personal conditions that were just discovered brought uncertainty, but never impinged on your motivation. I remember those days where you taught me a lot of courage, not to despair, to seek a solution with a positive attitude. Gabriele never saved a drop of sweat, would meticulously design and perform experiments tied to bold questions. Combining the skills of the two labs, you were able to show convincingly the benefits of marine-derived polysaccharides, from the beautiful Sardinian Sea to the only hills in the Netherlands. Gabriele brought actually a fresh vent of air, creating new vascular grafts with superior properties for endothelialization than any other clinical standards. Beyond the marine polysaccharides, you soon mastered different biofabrication technologies, combining them, making what today is a promising new concept for vessels that are hard to heal, such as, for example, coronaries. Let me also take this opportunity to thank the member of the assessment committee, as well as the defense committees at both universities, because you had to pass through the two different defense committees, for having taken the time to go through your thesis and be here with us today. In particular, I would like to thank Professor Pandit from the National University of Galway. Thank you, Abhay, for being always a source of inspiration, not only for the young scientists, but also for the somewhat more salt and pepper scientists like me. I would like to also thank Professor Bigun from Groningen. I'm looking forward to host you at Merlin Gosha, and I hope that we will have more and more chances to talk about new collaborations in the future. Professor Rotmans, Joris, thank you for your friendship and for the long-standing collaboration. It is a joy, actually, to be able now in these days to co-supervise Gabriele in his new journey as a not just started then officially postdoc in the new NICE project that we have together. To Dr. Crosio, Claudia, thank you so much also for all the nice time that I was able to spend many years ago when I was actually there as a visiting scholar, and when I actually had the opportunity to build also the long-standing collaboration with Antonio. And to my fellow colleagues, Professor Van Griesven and Professor Birgani, thank you, Nilofar, and thank you, Martijn, for your companionship. And Martijn, thank you for all the help that you have provided also to Gabriele along the journey in many of the hiccups and joys of the studies that he has conducted. I'd like to leave now the floor for a second, or for a few more seconds, to Antonio.
Okay. First of all, I would like to thank you, Lorenzo, for believing in our project and above all in Gabriele. Your mentorship and your companionship are invaluable to me. Dear Gabriele, son of Sardinia, one of the oldest and most fascinating lands in the Mediterranean Sea, shaped by thousands of years of history and an identity carved in stone. Its people are known for being reserved at first, but deeply loyal and warm once trust is earned. Gabriele, Sardinia is proud of its song. You have demonstrated that the finest science is not merely a matter of intellect, but of character. Your journey has been one of courage, determination, and will, and it has only just begun. Whatever the next chapter holds, I'm certain you will approach it with the same spirit that brought you through this one, steadily, consciously, and with a smile that puts those around you at ease. My warmest congratulations on the accomplishment achieved, and bright wishes for what lies ahead. Thank you.
We would like, therefore, to extend our congratulations to your family and all your friends from Italy and from Maastricht who kept you warm in all these years, worked with you patiently in the last bit of the journey, and listened to you and encouraged you in those discomforting moments that we all have had while finishing the PhD. Allow me some words in Italian, Mr. Prorector. Gabriele è un uomo molto educato, calmo, amichevole, e al tempo stesso con una grande tempra e forza di volontà. Gabriele ha dimostrato in questi anni come unendo lavoro sodo ad un innato ingegno si possano ottenere risultati eccezionali, per cui vorremmo ringraziarvi di cuore, a voi e a tutta la famiglia. Dear Gabriele, we wish you all the best for the coming years. Remember the lessons learned in these years in the Netherlands and in the special multicultural and international environment that is Merlin. We wish you to nurture more and more your leading capacity, as you showed us several times in these past years. And that when you put yourself into something, you start and finish it with outstanding results. Meneer de rector, ik heb gezegd.
Dear Dr. Obino, I would like to use now the official title which you have now carried. It's what we have in the Netherlands, our highest academic title which you have earned and which you have defended in a very gracious and very good way. In my congratulations, I also would like to bring forward your promotion team, so Professor Lepeda and Professor Moroni over here. And I think they guided you very well, especially when I heard from Professor Moroni that you did it officially within the restricted 4 years. Unfortunately, we don't have the incentive anymore, but this was otherwise, it was a special bonus. It's a double degree, and you had to defend it also in Sassari, as far as I understand. And that's also a compliment to you as well. I also would like to give my congratulations to your family who's sitting here, who came all the way from Sardinia, I suppose. And I think the candidate, your son, your family and brothers did a very good job. In my congratulations, no, in my thanks, I also would like to mention the members of the Corona who were here online, or they were from Leiden, or they were from Maastricht here, basically then, or from outside. I normally say it, they just have 7 minutes to ask questions, but it takes more than 7 minutes to ask these questions, to prepare, to read, and to digest the thesis itself very well. And by saying this, I think we're going to conclude officially here. For the conclusion, I use my hammer, which I have over here, as you can see, and I close officially this ceremony. How do we proceed? In 2 ways. First of all, we're going to make some pictures here, including the members who are online. We will guide you for this. We have a photographer here, I can see already here, the lady is over here. We have a reception in the so-called rafter. That's the place where we have some typically Dutch